Hello, everyone, and thank you for joining us for today's live webinar, Path Forward of Mass Spectrometry in Clinical Laboratories. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce today's speaker, Dr. Amy Chan, Associate Director of Point of Care, Department of Pathology, Anatomy, and Cell Biology at the Thomas Jefferson University Hospital. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Chan, you may now begin your presentation.
Thank you, Christy, for the introduction, and thank you very much for joining us today. The topic I'll be sharing with you today is the path forward with mass spectrometry in clinical laboratories. I just want to mention that I do not have any disclosure. So to begin, I will briefly describe the evolution of mass spectrometry application in clinical laboratories. The success of mass spectrometry in clinical application can actually be traced back to the 1980s, when it was primarily used to confirm drug testing. And during that time, it was discovered that there was a large number of false positive immunoassay results. Therefore, any positive antibody drug screen was considered presumptive positive until confirmed by gas chromatography mass spectrometry. Now, moving forward to the early 1990s, the development of the tandem mass spec for newborn screening was introduced. Now, this technology became very important in detecting congenital disorders in newborns, which later became a mandated screen in the U.S., although the specific tests performed vary among states. Now, as the new technology emerged in the mid-1990s, the development of MALDI combined with TOF, which is time of flight, and we will discuss that a little bit more in the next couple of slides, allowed for the rapid identification of microbes, which this later became FDA approved and widely implemented in the microbiology lab. This allows for decreased turnaround time compared to the conventional culture or biochemical tests. Now, fast forward within the last 2 decades, liquid chromatography mass spectrometry became popularly implemented within the clinical chemistry laboratories, measuring primarily small molecules such as drugs, vitamin D, and steroid hormones. Now, in the recent years, around the 2010, mass spec application has extended to protein and peptide analysis. With the development of more advanced technology, mass spec is now able to perform high-resolution analysis. Now, mass spec is not only became the gold standard for many clinical laboratory assays around 2013. Several journals, such as American Society of Endocrinology, implemented a requirement for mass spec analysis to publish steroid results. So this revolution of mass spec that we've just discussed is driven by continuous improvements in analytical platforms due to clinical needs, which are all anchored by the analytical specificity of mass spec. Now, what is mass spectrometry? So to put it very simply, mass spectrometry is an analytical technique that measures mass-to-charge ratio of an ion. Mass spectrometer produces large particles, or ions, from the chemical substances that are being analyzed. The mass spectrometer then uses electric and magnetic fields to measure the mass, or weight, of the charged particles. Now, I just wanted to very briefly go through the basic principle of mass spec. there are four main components of mass spec in the instrument, such as the sample inlet, which is not described in this figure, which is placed prior to the ionizer, the ion source, the mass analyzer, and the detector. First, the analyte of interest must be converted into ions. Then ions are separated by the electromagnetic field within the mass analyzer. Then it is detected by a quantitative method and processed into mass spectra according to the mass to charge ratio, which you can see on the right figure. The results are typically presented as a mass spectrum, which is a plot of intensity as a function of the mass to charge ratio. Now, there are very many different types of analyzers. The first one that I would like to introduce is the time of flight, TOF. The speed of which the ion travels for the TOF is actually dependent on the mass to charge ratio. And therefore, the lower the mass to charge ratio, the faster it will travel through the flight tube than ions of larger mass to charge ratio. So it's a very simple principle. Now, I just wanted to go over some of the applications of TOF. One of them is used in the detection of inborn error of metabolism. So on this figure that I'm showing is a profile of a newborn infant of normal metabolism profile. So what I would like to show you next is the profile of a child born with MSUD, which is maple syrup urine disease, which is an autosomal recessive metabolic disorder affecting branched-chain amino acids. Now, the newborn carries a genetic mutation in the gene complex that is essential for breaking down the amino acids, such as leucine, isoleucine, and valine. In this particular patient, there is an accumulation of leucine and isoleucine, which is very evident compared to the normal profile that I just showed you earlier. Now, the second application of TOF I would like to show you is in the use of drugs of abuse testing using urine specimen. In this case, this example I'm showing here is a multiplex system where you can detect multiple analytes in one run within one sample. They all have distinct mass to charge signature profile, and therefore we are able to separate each of them based on that. So you can see that there are several different drugs within this one specimen, and you can pretty much separate them individually and analyze them. The other common application of TOF is in the rapid identification of microorganisms at the species level by analysis of the intact ribosomal protein of the bacteria. The introduction of TOF in microbiology laboratory has significantly changed their workflow. MALDI-TOF mass spec is an innovative tool that is easy to use, rapid, accurate, and cost-effective, and it has revolutionized bacterial identification in clinical microbiology laboratories. The sample is co-crystallized with the matrix on the target and be desorbed and ionized by MALDI ion source. So in this figure here, you can see that once the source hits the target, it will be able to ionize the protein or matrix in this case, which are the ribosomal protein on the bacteria, and each spectrum will be matched will be read based on the structure of the ribosome and will be matched to a database profile to identify which microorganism it belongs to. The other analyzer type is triple quadrupole. This technology has been around since the late 1970s. Voltages applied to the pairs of electrodes set up an electric field that can cause ions to oscillate down the central axis as they travel through the device. The applied voltages determine which ions are able to make it all the way through to the detector, effectively filtering them. So essentially, one example I want to give you is, say, a compound A has a mass to charge ratio of 271. So when it goes through a triple quadrupole, which consists of Q1, Q2, and Q3 chamber, one can set filters detecting the specific mass to charge ratio that you would like to select for in Q1. So in this case, it's compound A, which has a mass to charge ratio of 271. Once it passes through Q1, it will get fragmented further in Q2 and therefore produce multiple daughter ions or product ions. And you can further select for specific daughter or product ions in Q3. In this case, we're selecting for 145 mass to charge ratio, and therefore you're able to increase the sensitivity and selectivity of what you're looking for. As I mentioned earlier, there is an increased number of assays being measured using MS over immunoassays due to its specificity, sensitivity, and its ability to build multiplex panel, meaning that one can measure multiple analytes in one run per sample. Some of the growing use of LC-MS in clinical chemistry include therapeutic drug monitoring, such as immunosuppressant drugs and antipsychotic drugs. Others include biomarkers such as vitamin D, steroids, and homocysteine. The use of clinical LC-MS has also extended to targeted protein quantification, such as thyroglobulin, IGF-1, renin, and angiotensin. The initial research work done on TDM, therapeutic drug monitoring, were lithium and phenytoin in the 1960s. At the time, researchers showed that there was a relationship between plasma concentration of phenytoin in patients being treated for epilepsy and the degree of seizure control attained. However, it wasn't until the 1970s that it became a routine practice in clinical laboratories, but with limited number of drugs being tested. However, between late 1970s and 1990s, there was a widespread use of commercially available homogeneous immunoassay, which resulted in the explosive growth in therapeutic drug monitoring because they were fast, cheap, and easy to develop. However, they're not without limitations in its clinical application, which we will discuss further in the next few slides. Since the early 2000s, the TDM trends began to move back to LC-MS to overcome some of the technical barriers, such as specificity and sensitivity. One of the issues regarding immunoassays on TDM is its specificity as well as sensitivity are sacrificed in certain assays. An example of this that I would like to use is methotrexate, which is a drug that is used to treat several types of cancer, as well as psoriasis and rheumatoid arthritis. It is a drug with a very, very tight therapeutic range. And some of the toxic effects of this drug includes it kills rapidly dividing cells, such as bone marrow, and it is highly nephrotoxic, and it can cause cirrhosis and under chronic low dosing. And therefore, oftentimes it is used in conjunction with rescue drugs such as leucovorin and glucocorticoids. One problem with using glucocorticoids is that it could cause some interference. So what is glucocorticoids? It is actually a bacterial enzyme that cleaves methotrexate and renders it inactive. However, during the cleavage, it could generate two non-cytotoxic metabolites, which are DAMPA, D-A-M-P-A, and glutamic acid. So in the figure in the middle shows the concentration of methotrexate measured by immunoassay versus LC-MS. As you can see, immunoassay overestimates the concentration of methotrexate in the beginning, the treatment. Now the reason is because, as you can see on the figure on the right, glucocorticoids, the structure of DAMPA is very similar to methotrexate, and because of that, there is a high cross-reactivity using the immunoassay antibody, and therefore you see a false elevation of methotrexate concentration, and it loses its specificity. And therefore, LC-MS is a better choice in terms of monitoring the concentration of methotrexate when patients are using glucocorticoids as the rescue. Another note to mention is that the accuracy, there's a huge effort in the standardization and accuracy project for immunosuppressant drugs that is commonly used in transplant patients. The evolution of medication for different targeted disease calls for a need to be able to measure its concentration in blood. So some of these are listed in this table. As you can see that a majority of these drugs, there are so-called new generation anticonvulsant drugs. A lot of them do not have immunoassay available in terms of measurement. and you can see that a lot of them uses HPLC or a mass spectrometry method to measure the concentration in blood. These days, you can measure a panel of drugs in one run per sample. This figure elegantly shows the separation of a 20-drug panel in 7 minutes. Now, each spectrum here represents each drug within one sample. TDM has also extended to measure antifungal drugs such as triazoles. Such ability to measure multiple analytes brings value to those patients with neutropenia either caused by malignancies or chemotherapy, bone marrow transplant recipients, and or other solid organ transplant recipients, because they are commonly taking multiple antifungal drugs at the same time. So this is just a figure showing that a patient who is taking multiple drugs at the same time. So you can measure that there are different concentration with very nice separation using mass spec. Other therapeutic drug monitoring that uses mass spec also includes other anti-neoplastic drugs such as busulfan and therapeutic metabolites. Here, I wanted to discuss the hormones measurements by LCMS, which is becoming a demand among the endocrinologists. The measurement of hormones, especially steroidal hormones, requires high sensitivity and specificity measurement. This is particularly important for low-end sex hormone measurements in the context of a particular hormone in patients suspected of sex hormone disorders, which are listed in the table. Dihydroxyvitamin D is also commonly used to be measured by LCMS. This is due to its picomolar concentrations and its lipophilic nature, and therefore there are some immunoassays depending on the vendor that you use. It's not very standardized, and you get varying results using immunoassay method. Another common hormone analyzed by mass spectrometry is free T4, because thyroid hormones are usually highly bound, protein bound. The gold standard for free T4 is actually equilibrium dialysis. Now, one limitation of equilibrium dialysis is that it is very laborious. It takes a long time to perform this test. By using LCMS, you could get a faster turnaround time. I just wanted to point out the importance of hormone measurement by LCMS by using estradiol as an example. In this case, as you can see on the left panel of the table, the limit of quantitation of a direct immunoassay is around 20 to 30 picograms per mil. Now, if you extrapolate this to the figure on the right, one can only capture a specific population of condition, such as women receiving gonadotropin therapy or premenopausal women. However, as you can see, there's a subpopulation of patients which are below the detection limit of a direct immunoassay, and one could miss diagnosis. So with mass spectrometry, one could measure as low as 1 to 10 picogram per mil of estradiol. In this case, you can see that how it is important to be able to identify this subpopulation of patients that could be missed if using immunoassay-based method. So there was a position statement that was put out by the Journal of Clinical Endocrinology and Metabolism back in 2007. Essentially, they were evaluating the current state of clinical assays for total and free testosterone. What they found out was that laboratory proficiency testing should be based on the ability to measure accurately and precisely samples containing known concentration of testosterone, not only on agreement with others using the same method. So at that time, there was a lot of variations in the measurement of testosterone using different platforms, and they feel strongly that in order to be able to diagnose the patients properly, there should be a consensus and to be able to measure them accurately. And this is followed by another position statement that was put out in 2013, which is very similar, but this is in regards to estradiol. And therefore, a consensus, and there was a consensus that came about where if you were to publish any steroid hormones, specifically sex hormones, in the Journal of Clinical Endocrinology, one needs to use a mass spec-based method to be able to, that is essentially one of the requirements to publish a paper. So I just wanted to, as I mentioned earlier, one of the application of mass spectrometry in clinical labs is protein analysis, which is becoming more and more important and common in clinical laboratories. Some of the examples include thyroglobulin, IGF-1, amyloid protein, plasma renin activity, aldosterone, and insulin. So I just wanted to point out that there's a reason why it is becoming more and more important to measure thyroglobulin by LC-MS. But before I talk about the difference, let me discuss a little bit about why thyroglobulin, what are its utility in clinical setting. Thyroglobulin is the primary biochemical tumor marker used to detect recurrence in patients with differentiated thyroid cancers. The problem is that the results can vary depending on which immunoassay platforms that you use. So there is no standardization in terms of this assay. And it is found that 20 to 25% of the patients with differentiated thyroid cancer have anti-thyroglobulin antibody. So when we deal with immunoassay, one caveat is that when a patient produces anti-thyroglobulin antibody, it could interfere with the measurement of thyroglobulin because it could bind to the reagent of the assay. And therefore, you will not get a very accurate measurement. So this is a study that elegantly shows the effect of anti-thyroglobulin antibody with the thyroglobulin immunoassay. So on the figure panel A, what we're looking at is patients with differentiated thyroid cancer with thyroglobulin antibody negative. So in these patients, one should assume that they do not have any interference in their immunoassay because they do not generate thyroglobulin antibody. And as you can see in the black solid line, a majority of the patients, when compared to using immunoassay, which is the first bar column, versus LC-MS, second bar column, versus a gold standard, which is radioimmunoassay, third column, there is no significant difference in terms of their thyroglobulin measurement. However, as you can see that there are some red dots, those are just a subpopulation of patients. There were only a handful of patients that do not correlate at all for some unknown reasons. And if you look at the right panel, panel B, these are differentiated thyroid cancer patients with thyroglobulin antibody positive. And you can see that when comparing using immunoassay versus LC-MS versus radioimmunoassay, a majority of these patients, their thyroglobulin level is measured lower compared to the conventional and gold standard method, which are LC-MS and RIA. So this strongly suggests that the presence of thyroglobulin antibody interfere with the measurement of thyroglobulin, so therefore could present as false negative in terms of cancer recurrence, so it became problematic. Now, I wanted to switch gear to discuss a little bit about the utility of LC-MS in monoclonal antibody therapeutics. If you recall at the beginning of my presentation, LC-MS has the capability to analyze peptides and proteins. Now, with the boom of immunotherapy for cancer treatment, monoclonal antibody therapeutics are now widely used. There are currently more than 20 monoclonal antibodies that have been approved by the FDA. However, there are no assays available, or there were no assays available to measure the efficacies of this drug in patients until the development of the method using LC-MS. So essentially, this graph here shows that the association between the serum concentration of infliximab, which is a monoclonal antibody, and its efficacy in patients with ulcerative colitis. So as you can see here, patients who respond well to the treatment, they have a higher circulating concentration of infliximab in their blood compared to patients with no response. So this allows for physicians to be able to see whether or not the drug is effective on the patient or whether or not they need to adjust the dose of this therapy. Now, without going into absolute detail on how one can use LCMS to detect monoclonal antibody, but essentially, if you look at the graph on the left, at the bottom, you can see that we could differentiate infliximab with adalimumab in human serum. And on the right-hand side, you can see that this peak, we know what this peak belongs to due to its signature peptides. So essentially, we can generate signature peptides from the protein of interest to perform targeted protein analysis and thereby being able to quantitate its absolute concentrations. What does the road ahead of us look like in terms of adoption and expansion of mass spectrometry? So some of the challenges that we face in the lab in terms of adopting mass spec-based assays include access to the equipment. The capital cost for the equipment is not cheap. It is around $200,000 to $500,000 per piece. Also, it involves training of skilled personnel. This test, mass spec, is considered a high complexity testing. In addition to that, mass spec is also considered a laboratory developed test because they are not FDA approved. That means that one will require additional validation before an assay can go live. So what are the drivers for continuous expansion for mass spec-based assay? One of the most important one is certainly the clinical needs in order to, that I presented and I've talked about earlier, some of the importance of accurate hormone measurements, steroid measurements, and vitamin D as well. And also high throughput instruments. So currently, which I'll discuss a little bit later, mass spec assays are not as high throughput compared to automated immunoassay. However, there are works to make them high throughput by multiple vendors so that they can perform in more volumes to decrease the cost. The other part is also information technology and vendor support. Currently, they are not as well supported to the conventional automated platforms. However, it's been evolved. There's been a lot of discussion between vendors and laboratories to improve those processes. So, I just wanted to quickly go through the workflow and challenges of mass spectrometry, how it can be manual, laborious, and involves multiple workstations. So just quickly, when we receive the sample, one has to process the sample by adding internal standards, which is sort of an internal control. And then somehow there would be involving a protein purification step, either it be SPE or protein precipitation. And then the technologist would have to load those samples onto the autosampler, which the probe would individually sample each samples. And then it would have to go through the HPLC sample pump and LC column, which are the main separation technology or the process that occur. And then finally, it would flow through into the mass spec for analysis. And each individual process should be monitored tightly, and it's still a very manual process, even though some parts can be automated. But there are softwares that can do analysis once a spectrum is generated. So these are FDA approved, where it incorporates chromatogram analysis, peak integration, and generation of a calibration curve. As I mentioned, it is a multi-step process, and therefore one should establish a sustainable workflow. So therefore, we should include technologist training on operation and result analysis. So I would suggest preparing a checklist of things, including proficiency testing, training, daily run information. So tracking daily run information should include sample preparation, such as cartridge lot differences. Make sure that we log in the individual, catalog individual buffer lots, open date, and expiration date, since these are all manual made components. And also test run, batch information, mobile phase, column pressure, mass spec parameters, all should be tracked daily. And data analysis is also an important part to monitor the internal standard. Again, something that I mentioned earlier, it's an internal control. We wanted to make sure the retention time of the same analyte you're measuring always come out at the same time. You want to check the peak shape and the analyte area, making sure that they're consistent. And then one thing is also to design a troubleshooting workflow. So as I mentioned earlier in the previous slide, it involves so many components, and any component could malfunction. So it would be helpful to design a troubleshooting workflow for the technologist to go about troubleshooting if they see an increased pressure, where should they look at, and whatnot. And currently, with the tech support from the vendors are not established as 24/7. So in order to have them come and take a look at it might take a day, and therefore it's easier to have the technologist to perform initial assessment of the issue. It is also important to note that one should have a performance program, monitoring program, and internal audit program. So essentially subscribe to CAP-PT, proficiency testing. If no proficiency testing is available, one should do an internal exchange with another lab that performed the same test to ensure that you're getting consistent results. So how can we improve the workflow? And I mentioned that it's very manual. Some of them can be improved using sample preparation. The process can be improved. We could adopt a liquid handler platform where the liquid handler is an automated platform. It could automatically pipette a fixed amount of buffers, a fixed amount of washing buffers and reagents. So you would take out the manual part. I know that there are several vendors that have currently developed an automated analyzer that is very similar to the chemistry automated analyzer. I believe it is currently being sold in Europe. However, they are not currently FDA approved to be used in the United States. They did, however, develop several kits. I believe the vitamin D kit is one of them, where you can buy a kit and you don't have to develop the test. It's already an approved test. What you do is just put in little bottles. It will have all the parameters set. You just have to push one button and it would analyze the sample, just like a regular chemistry automated analyzer. Now that I've talked about the workflow, I just wanted to use the leftover time to talk a little bit about the expansion application of mass spec in terms of the current state of it. What is it currently used in research? So clinically, Mayo Clinic has developed an assay that allows for diagnosis of monoclonal gammopathies. So just briefly about monoclonal gammopathy, it's a disease of the bone marrow where it generates monoclonal malignant plasma cells, monoclonal immunoglobulins, which are then secreted into the bloodstream. The current conventional method is using gel electrophoresis. At the Mayo Clinic, they have developed an assay where they can analyze the monoclonal antibodies within the bloodstream and quantify them, as you can see on the right-hand side of the figure. So as I mentioned before, the gold standard currently is using gel electrophoresis. However, gel electrophoresis has its own limitation as well. For example, it is very subjective to the reader because it's a stained gel. One might see a band and one might not see a band when it's faint. Therefore, there is a need for improvement in terms of quantification, since this is some semi-quantitative using the current method. So what Mayo Clinic have done is that they have created a mass spectrometry called a mass fix that is able to analyze a wide variety of M proteins, and they can identify the proteins by their heavy and light chain, as shown on figure on the right. And they can even quantitate the protein level in terms of concentration. So the next exciting part about the use of mass spec is in the measurement of functional proteins on formalin-fixed paraffin-embedded tissue, FFPE. And now this is under research. What you can do is that you take an FFPE tissue and perform microdissection of the piece of tissue, and then process the tissue and digest the tissue and subject it to quantification detection within the mass spectrometry. The reason why we're interested in using mass spec to perform protein quantification is because the conventional method using immunohistochemistry is again using antibody. So some of the antibody-based assay is not very specific or sensitive, and not all analytes or all biomarkers have antibodies available for performing IHC. So in this case, you can get an absolute quantification whether or not a patient expresses certain biomarkers on the tissue cell surface. So I wanted to point out that one of the clinically implemented case of mass spectrometry assay is on amyloidosis, which is currently in clinical use. It is the identification of the type of amyloid deposited is critical to the correct diagnosis and treatment of patients with this illness, and therefore characterization of systemic amyloid deposits by mass spectrometry is already clinically implemented in several hospitals and reference laboratories. Some of the biomarkers that are currently in research include the breast cancer marker HER2 and the immune checkpoint PD-L1, which was actually presented at one of the poster session, which is a collaboration between MD Anderson and University of Chicago. So this is the end of my presentation, but I just wanted to quickly summarize some of the take-home messages from my talk. And I hope that the audience can appreciate how far we have come in terms of using this technology that's been around for decades, and how we are able to advance medical diagnosis with its implementation in clinical laboratories. Now, it is certainly not, it does not come without some limitation. There is this upfront cost that we talked about, how it can be a big capital for some institution, and it also involves personnel training and method development and establishing a sustainable workflow. But I really believe with increased demand and practices calling for standardization, there will be a push for an approval of an automated system in the U.S. Now, it might take some time to be able to formulate kits for each assay, but I think there's certainly a market for it. So with that, I would like to thank the audience for listening in and participating in this webinar, and I'm open for live questions.
Thank you, Dr. Chan, for your informative presentation. We will now start the live Q&A portion of our webinar. Now, we already have some great questions coming in, but as a reminder to our audience, if you do have a question you would like to ask Dr. Chan, just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. Dr. Chan, let's go with this first question. What resources are needed to establish a clinical mass spectrometry laboratory?
Hi, that's a really good question. So one would start with the capital requirement for purchasing an equipment. So as I mentioned earlier, it usually costs about $200,000 to $500,000, depending on the resolution of the mass spec that you are looking at, and of course the lab space. One would make sure that there's enough lab space to put this equipment. It's a pretty big equipment. And the other one is clinical need assessment. Is there a clinical need? For example, I talk a little bit about some of the immunosuppressants that can be used to measure using mass spectrometry. And is there a need? Do we have a big transplant program? What is the volume to bring in specific tests? If we're sending out some esoteric test, do we have an increased volume that it could justify the cost to bring it in-house? So these are some of the things to consider, and certainly not to forget. Training of qualified personnel to perform high complexity testing is important. It does take some time to train a personnel to be competent at performing this very high complexity testing. In my personal experience, it sometimes takes 4 to 6 months to train somebody without any experience. So certainly consider all these points to establish a successful mass spectrometry lab.
Thank you, Dr. Chan. Our next question, since most MS assays are lab-developed tests, how do you maintain and ensure test quality?
Yeah, so it is important to establish performance monitoring program by tracking daily data analysis, such as running QC, monitoring the internal standard, looking at the retention time, making sure that what you're measuring elutes out at the same time all the time, so that we know that it's consistent, it's the same analyte you're looking at. We want to look at the peak shape, making sure you track the pressure. And then the most important thing is establish an acceptable threshold. What is considered acceptable? Now, the lab director will have to establish this because this is a lab-developed test. There's no necessary guidance from any regulatory body. It is also important to catalog and track lots when you open a new lot of certain reagents, and make sure that you put in an expiration date and perform lot-to-lot comparison in patients. Every time you change your lot, you make sure that you perform lot-to-lot comparison, making sure that the same patient still measures the same despite changing the lot. And it is also important to perform internal audit program or external audit programs, such as subscribing to CAP proficiency testing, or alternative assessment program when one cannot subscribe to PT because they don't have it. So you would want to exchange specimens with other laboratory performing the same test, making sure that you establish an acceptable number or limit of error of what you're looking at.
Thank you. For MS analysis on proteins, do you pre-treat samples to increase sensitivity and specificity? And if yes, how do you do that?
Yeah, this is a really good question. So for protein analysis, especially in matrix such as the whole blood or serum, it is a good idea to pre-treat the proteins. So for those who are not familiar with this, pre-treating means that if you have a blood or urine or some specimen that consists of many different kinds of proteins in there, how do you purify them to increase sensitivity and specificity? So it is very common for when they develop this test to use a monoclonal antibody to bind, or sometimes people call it to fish for or to enrich for a particular protein of interest that you're looking for. So therefore, once you have a monoclonal antibody that binds to the protein of interest, it would precipitate. So therefore, you can increase the selectivity, sensitivity of your assay. Another thing is to perform albumin depletion, because most of the specimens consist of high percentage of albumin. So if you can pre-treat it with albumin depletion treatment, you can get rid of abundant non-targeted proteins, and therefore you can increase sensitivity and specificity of your assay.
Thank you. Now we have time for just a few more questions, but I want to remind our audience that those questions we are unable to answer today and those that come in during the on-demand period will be answered by our speaker via the email address you provided at the time of registration. Dr. Chan, what are some of the limitations of the MALDI-TOF in the identification of microorganisms?
Yeah, so again, the top limitation is, of course, the initial high cost of the instrument, but the technology itself in terms of limitation, is the identification of new isolates is possible only if the spectral database contains peptide mass fingerprints of the type strains of specific species or subspecies. So if the library or your database doesn't have that, you cannot match it once it produces or generates a spectrum, then you can't really release the result because it's not in the database. But however, I do know for sure that Mayo Clinic has generated their own database where they collect different strain and species of microorganism and start building their own database that they commonly see. Therefore, they have actually a large library compared to the standard ones that we get in the lab from the vendor.
Now, our last question. You mentioned mass spectrometry replacing gel electrophoresis. How does one bill for the test? Does it have a CPT code?
That is actually a very excellent question. So that I myself questioned that also. So currently there is no CPT code to order the mass fix assay, which is the mass spectrometry method of analyzing the monoclonal antibodies for monoclonal gammopathies. I do know that Mayo Clinic currently offer that to the patients who are currently seen at the Mayo Clinic. In terms of how they get charged, I'm not so sure. We will have to ask them. And I do know that if you were to order it from a reference standpoint, say, I don't have this test, I want to order it from Mayo, how do we get billed for that? That I do not know. I've never sent it to them before, but I do know is that they have submitted an application to CMS to apply for a CPT code. So it would be interesting to see by next year or so if there would be a code for them, and we would know how to charge our patients, because I think that it will become more of a standard practice to use that technology in terms of measuring monoclonal gammopathies instead of gel electrophoresis.
Thank you, Dr. Chan. Do you have any final comments for our audience?
No. Is there? I'm very excited to share this discussion with all of you, and I enjoy presenting this presentation. Thank you for the opportunity, LabRoots.
Thank you. And I'd also like to thank our audience for joining us today and for their interesting questions. One last reminder that those questions we were not able to answer today and those submitted during the on-demand period will be addressed by Dr. Chan via the email address you provided at the time of registration. I would like to once again thank Dr. Amy Chan for her time today and her important research. We'd also like to thank LabRoots for bringing you today's live webinar. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us. Have a great day.